Hello students, in this video I am going to explain about the digestion of food in small intestine by pancreatic juice and bile. Various secretions like bile, pancreatic juice, intestinal juice, are liberated into the duodenum part of small intestine. In the small intestine, these secretions are thoroughly mixed with the food by the movements that are generated by the muscularis layer. So this mixing of food with these juices is facilitates for the digestion of food. Bile and pancreatic juice so these juices are liberated through the duct, that is hepatopancreatic duct. So it is important to remember this point, from which duct bile and pancreatic juices are liberated. Answer is hepatopancreatic duct. Now let us see the constituents that are present in the pancreatic juice and bile. Pancreatic juice contains various enzymes like trypsinogen, chymotrypsinogen, procarboxypeptidases, and amylases, lipases, and nucleases. And it is also having some bicarbonates. So these all are the constituents that are present in the pancreatic juice. Now let us see the constituents of the bile. Bile is having bile pigments, they are bilirubin and biliverdin. So these are known as the bile pigments. Bile salts, cholesterol, and phospholipids. So these all are the constants that are present in the bile. But enzymes are absent in the bile. No enzymes are there in the bile. So it is very important point to remember. So you are also supposed to remember the constituent of the pancreatic juice and the constituents of the bile. So these bicarbonates are helpful for the providing alkali pH. So that is 7.8. So this alkali pH is helpful for the activation of the some of enzymes. So this trypsinogen, chymotrypsinogen, procarboxypeptidases are inactive enzymes. Trypsinogen activates into trypsin. In the presence of an enzyme that is enterokinase. So enterokinase enzyme was liberated by the intestinal mucosa. So it is important to remember this point. Trypsinogen was get activated into trypsin in the presence of enzyme that is enterokinase. And enterokinase enzyme was liberated by, answer is intestinal mucosa. This activated trypsin further act upon the remaining inactive enzymes and get them to the activated. Chymotrypsinogen activated into chymotrypsin. And procarboxypeptidase enzyme activates into carboxypeptidase enzyme. So these two reactions occur in the presence of trypsin. And actually protein digestion started in the stomach. So proteins are partially digested. These partially digested proteins next reaches to the small intestine. They acted upon by the pancreatic juice. And here are the enzymatic reactions of the pancreatic juice. This activated trypsin converts the proteins, proteoses, and peptones. So these all are the partially digested proteins get convert into dipeptides. In the presence of trypsin or chymotrypsin or carboxypeptidase. And polysaccharides converts into disaccharides. in the presence of enzyme that is amylase, pancreatic amylase. And next, nucleic acids converts into nucleotides and then to the nucleosides. In the presence of nucleases. Now, bile salts emulsifies the fats and break them into smaller micelles. So this reaction happens in the presence of bile salts. So emulsification means breakdown of the fats into smaller micelles. So it is very important. So emulsification of the fats done by the bile salts. And this bile also activates the pancreatic lipases to act upon the emulsified fats. So these emulsified fats converts into diglycerides and then to monoglycerides. So this happens in the presence of activated lipases. So this is about the digestion of the food by the pancreatic juice and bile. So thank you, students. If you like this video, please subscribe my channel.